Next, we will move on to the final speaker of this session. We will hear a lecture by Dr. Yosuke Togashi, Professor of the Department of Tumor Microenvironment, Faculty of Medicine, Dentistry and Pharmaceutical Sciences at Okayama University. Dr. Togashi, please come up to the stage.
Thank you for kind invitation and introduction. I'm Yosuke Togashi from Okayama University, West Side in Japan. Today my talk focuses on cancer immunology, especially to mitochondrial transfer in the tumor microenvironment. So I'm a medical doctor majoring in pulmonary medicine, respiratory medicine. So we are very impressed by cancer immunotherapy, especially PD-1 blockade therapy. So advanced non-small cell lung cancer are resistant to any chemotherapy more than 10 years ago. However, after cancer immunotherapy era, some patients could achieve durable response similar to cure. But the frequency is less than 20%. So we want more patients to achieve such durable response. So 10 years ago, I started cancer immunology research in Professor Nishikawa's laboratory as postdoc fellow. Professor Nishikawa's laboratory originated from Professor Shimon Sakaguchi's laboratory. So my first project mainly focused on regulatory T cell. So we found PD-1 was highly expressed by tumor-infiltrating regulatory T cell. And PD-1 blockade activates such PD-1 positive Treg cell-mediated suppressive function, leading to resistance to PD-1 blockade therapy. This article published more than 5 years ago in the collaboration with Professor Sakaguchi and Professor Honjo. Because I graduated from Kyoto University Medical School, so it's my great experience to collaborate with a huge giant, 2 Nobel Prize winner. When I entered into Kyoto University Medical School, Dr. Honjo was the dean, and Professor Sakaguchi also Kyoto University Medical School professor, so very exciting work. After publishing such data, such study, I received several offers from other institutes to have my own laboratory. At that time, I wanted to have my original research project in addition to regulatory T cells. So I was also interested in effector T cell, especially tumor-specific T cells that attack tumor cell directly. Because PD-1 blockade activates such T cell, and such T cell attack tumor cell, leading to tumor regression. So we analyzed the tumor-infiltrating lymphocyte from patients treated with PD-1 blockade therapy by the single cell level to identify tumor-specific T cell. So this is conventional, traditional single-cell analysis data, flow cytometry data, CD3 and CD8, just 2 parameters and just one such scatter plot. For example, this population CD3 positive T cell, and this population CD8 positive CD3 positive T cell. And this population may be CD8 negative, meaning CD4 positive CD3 positive T cell. So this is traditional conventional flow cytometry analysis. However, more than 30 parameters need more than 500 such scatter plot. It's impossible for us to analyze such data. So we use bioinformatics analysis technique, seeing the believing. I show demonstration. Each dot means immune cell, and the same immune cell, similar immune cell gathered cluster, making cluster island. We call such island cluster. So such cluster can be created by automatically. So, for example, this cluster is CD8 positive T cell. This cluster is CD4 positive T cell. So using such technology, we analyze the TIL from actual patient treated with PD-1 blockade therapy. We focus on this cluster. This cluster was dominant in responder to PD-1 blockade, and this cluster was not dominant in non-responder. This cluster was a very simple cluster, PD-1 positive, CD8 positive T cell. CD8 positive T cell, we so-called cytotoxic T cell, main character in anti-tumor immunity. And such PD-1 positive such T cell infiltration could be predicted biomarker from our data. And we also sorted this population, and we performed the immunological assay to respond to tumor cells. So we confirmed such PD-1 positive, CD8 positive T cell actually attack tumor cell. We call such T cell exhausted T cell, PD-1 positive exhausted T cell. So in my own laboratory, I started exhausted T cell project. Exhausted T cell have several features. For example, immune checkpoint inhibitory molecule highly expressed in exhausted T cell, or epigenetic change occur in exhausted T cell. So in addition to such feature, I'm interested in metabolic dysfunction, mitochondrial dysfunction in exhausted T cell. What is mitochondria? Mitochondria are very famous organelle and produce ATP energy, cell energy, through oxyphos. We call such a metabolic state respiratory oxyphos. And mitochondria was more than 1 billion years ago. Mitochondria was bacteria, ancient bacteria, and such bacteria enter into eukaryotic cell and became organelle. This is endosymbiotic cell theory. So according to such theory, such mitochondria can transfer from cell to cell. This is mitochondrial transfer research field. 20 years ago, mitochondrial transfer was first reported in experimental model in PRONAS, but there is no evidence in human clinical sample. So 5 years ago, when I searched for my own original project, and I met this phenomenon by chance, so I started mitochondrial transfer project in the tumor microenvironment. We first analyzed mitochondrial DNA. Mitochondrial DNA, original DNA, different from nuclear DNA, because mitochondria originally bacteria. So such bacteria had original DNA. So we first sequenced mitochondrial DNA using clinical sample, especially TIL, paired cancer cell, and peripheral blood. So very lucky, we found mitochondrial DNA mutation in TIL, 40% TIL, many of which shared with paired cancer cell. This may be from cancer cell to T cell transfer. So we conducted a transfer experiment. We stained TIL with green color. We stained TIL mitochondria green color, and stained cancer cell mitochondria with red color, and co-culture for a couple of days. 2 or 3 days later, red color enter into green color and merged, change yellow color, meaning mitochondrial transfer in the tumor microenvironment. What mechanism of mitochondrial transfer? There are several reports about mitochondrial transfer mechanism. Please look at these bar. These bar we use insertion column, separation column during co-culture. Such insertion column inhibit mitochondrial transfer. In addition to insertion column, this inhibitor also inhibit mitochondrial transfer. This inhibitor is nanotube inhibitor. So tunnel nanotube attached with cancer cell and T cell induce mitochondrial transfer from our experiment. In addition to tunnel nanotube, please look at this bar, this column. We use this inhibitor in this condition. This inhibitor is EB inhibitor, exosome inhibitor. So exosome, small extracellular vesicle, also play an important role in mitochondrial transfer. We also analyze function of T cell after transfer. Please look at these bold letters bar, shadow bar. These bar mean mitochondrial DNA mutated to T cell line. For example, mitochondrial respiration oxyphos inhibit by mitochondrial DNA mutation. And dependency on glycolysis increase opposite metabolic state respiration and glycolysis, and dependency increase in mitochondrial DNA mutation. So mitochondria metabolism very important for T cell memory formation. So we analyze the memory formation in T cell function. For example, central memory and long-lived memory T cell. Please look at the red color shadowed bar. Red mean transferred T cell data, and shadow mean mitochondrial DNA mutation transfer. So, for example, central memory T cell clearly decrease, and long-lived memory T cell also decrease. We also use PD-1 and CD69 as activation molecule, and PD-1 and CD69 also decreased after transfer, mitochondrial DNA mutation transfer. We also created this conditional knockout mouse. This conditional knockout mouse has T-cell specific mitochondrial dysfunction. And please look at the red line. Blue line control mouse and red line conditional knockout mouse. PD-1 blockade resistant sensitivity decrease in this red line. In addition to just the efficacy of PD-1 blockade, we also checked memory formation in this model. After PD-1 blockade, several mice completely reject tumor cells. And we corrected such rejecting the mice, and more than 1 month later, no PD-1 blockade therapy, no PD-1 antibody in this mouse, but we rechallenge in such memory mice, same parental tumor. Blue line control, all mice reject parental tumor, memory formation very fine in this mouse model. However, in this conditional knockout mouse, just one third rejected and 2/3 tumor grew rapidly. So memory formation in this mice were impaired, clearly impaired. Finally, we also sequenced the clinical sample, about 200 clinical sample from melanoma patient and non-small cell lung patient who received PD-1 blockade therapy. Red line mitochondrial DNA mutated patient and progression-free survival and overall survival tend to shorter in mutated patient compared to wild-type patient. So we concluded tumor cell-derived mitochondria mutated transfer to immune cell impair anti-tumor immunity and ICI efficacy impaired in such state. So we published this data in Nature last year. We also analyzed cytotoxic chemotherapy efficacy, no change, mutated and wild types. So this tendency was specifically observed in ICI therapy. I appreciate every collaborator, especially Nature First Author, Dr. Ikeda, my PhD student, and final MyeloCell project, Dr. Tanabe and Dr. Omae, my present PhD students. Thank you for your attention.
Thank you very much, Dr. Togashi.